Can you see it now, all right?
Yeah.
Thank you. So thank you very much indeed. It's a great honor to be here, as always. And this is a very special commemoration with some familiar faces there of the wonderful time we had visiting Sweden. Amy and I, Amy is on the call. We're now in Australia, but still comes back to the UK to work on Gaucher with us. And that's a wonderful experience we had with the very famous family that you all know. Thank you, Bosse, for looking after us so handsomely with your colleagues. So I wanted to give a personal overview of the disease very quickly, but just to kind of celebrate and to just show you, or just to think what might be happening before Amy talks about our local project, of which she's brought so much, and which is going to bring a lot more later. It's been 7 years really since we were with you. And I know he's Danish and not Swedish, but Kierkegaard did say this, which is life can only be understood backwards, but must be lived forwards. And that's what we're doing now. And that's what we've got to do. So an overview. It's been 1881. Okay, sorry, connection's unstable. I can't help that. 1881. And we have learned a lot from this time of Gaucher himself. We've learned the severity of the disease more even than he knew, in the bones, in the viscera, and also, of course, in the brain. And the brain and the bones remain important things to conquer. We know that the cells that are found in the spleens and liver are macrophages. And we know that in those macrophages, there are distended lysosomes containing a material that was identified also in France, first of all, in 1934, moved on 50 years. And we know that the multi-system nature of the condition reflects in part where macrophages are, but also just the way the disease is in the brain and the bones, and of course in the viscera. That's what macrophages do. And understanding that was crucial for understanding the therapy. First of all, that the abnormal macrophages, particularly studied here in Sweden by Maciek and colleagues at the Karolinska, has shown again such important images for us to know today, and that this lady identified this material, and understanding this alone made huge differences in developing enzyme studies, understanding the defect, and indeed beginning to tackle it. And in essence, that simple approach there has given us cell replacement, bone marrow transplantation. It has given us a vision of a particular type of gene therapy that Professor Carlson has developed more effectively, and indeed twice in his professional life, that's coming into clinical application. It has brought us enzyme therapy. It has brought us substrate reduction therapy, and it has enabled us to think about all aspects of this model condition, which is very rare, very severe, and yet a model for huge human success. And this image, which is a very special image known to you all, is something we don't forget from our visit in the family that formed your association, and indeed the early treatment by bone marrow transplantation, as it then was by Professor Ringen, and with Dr. Svennerholm, Professor Svennerholm, who pioneered this from Sweden. So Sweden has tremendously important contributions in its record of achievement. We have in our cohort and known to Amy, a patient who's now age 49 a man treated in the very same year who is still going strong after a very serious illness from 18 months of age and a treatment by marrow transplantation many years later when he was about 30. So we have to remember that he was part of a cohort that Amy with Mechit is writing up of patients who have received this treatment and reviewing its success actually over many years, showing these are some patients written up from the UK, but the Swedish and the UK series is the biggest one in the world and contributes a lot to our understanding. You know about enzyme therapy developed all those years ago, 20 years of development, knowing the enzyme defect by Roscoe Brady, who died a few years ago, a courageous and determined and brilliant man who showed a lot of strategic thinking. His partnership with Henry Tamiya and the Gaucher's Association of America, the National Gaucher Foundation, was important for bringing about those trials of enzyme therapy from this single patient, Brian Berman, who triumphed for children and a realtor, as they say, in New York. And this was the enzyme therapy. I'm sorry, my connection is unstable. Sorry for that. This is the enzyme therapy. And here it is, approved with a recombinant form later. And we know after 10 years, after 20 years, in follow-up studies done by the company that pioneered the first therapy, there are others, that this therapy is effective for the systemic manifestations of the disease, provided the patient gets it early enough. Here's the 20-year data. It's maintained at 20 years for almost all the outcomes that are easily measured. Best to have the treatment young. And it depends on how bad the disease, of course, is before you start it. Doesn't work for the neurological condition, but helps patients a lot. And the first patient in the UK was treated in 1991. So it's 31 years, and he's still going strong today and was able to go back to work having been in a wheelchair. This young lady is like the modern patients today with type 1 disease. She was treated very young. She was thought to have a terrible blood disease. And there she is, a working lawyer with 2 young children, celebrating the 70th anniversary of the National Health Service. And is very grateful for everything that has gone on. She's one of a cohort from them. This is the complicated biochemistry of the lysosome, which can be delivered to with enzyme therapies. It can be understood from the point of view of replacement for whole cells that where the lysosomes are deficient. But it acquires the sphingolipid, the glucosylceramide, from outside, and it's controlling the supply from outside, which is another means of therapy. That's what it does. And so the evolving therapies have involved then the idea also of restricting the abundance of that material, its manufacture, like one would give a statin or hypercholesterolemia for people who have too much cholesterol, and the storage material, if you will, in their blood vessels. So it is that we have thought of substrate reduction therapy in this condition. The first treatment, which was identified in preclinical studies in Oxford, was something that I tried out in Gaucher disease type 1, a non-neurological form. And that was in 1996 to 2000. And it had an effect. It kind of worked. It restricted the oversupply, is the idea of statins, and it controlled the oversupply and enabled the still functioning, but not very effective enzyme digestion in the lysosome, it enabled it to cope and prevent the overproduction, the storage of this material, which is the source, we're pretty sure, of the disease. Another drug, more effective than miglustat, the first approved one, which is used in Niemann-Pick disease, but not Gaucher very much today, is eliglustat. And that was developed in America through the efforts in the late 90s and early 2000s by Jim Shaman, following on the efforts of Norman Radin at Ann Arbor University at Michigan, University of Michigan, US, where the cars are made, in Detroit. So this was approved. We did some of the early trials of this in 2014 and 2015, and is an orally active treatment just for the peripheral manifestations. But it is like an enzyme therapy, as you see here in the phase 2 studies, very effective for the blood abnormalities, the liver and spleen enlargement. And you can show in a controlled study that it has good effects, and good effects that are maintained. We now know, and this has not been published, but is about to be submitted for publication, is that it, sorry, my connection is unstable, but that it has an effect on the thin bones, particularly in the vertebrae, as shown here in patients seen in those first trials over a period of some years. You can see that over years, thin bones that are susceptible to fracture and painful crush fractures are improved by this therapy, perhaps because a soluble compound can penetrate the marrow in difficult places like the vertebrae and have its effect on the local cells there. The bone marrow burden of abnormal marrow also improves in years on this drug. In the 2 studies that I've been involved with most, you can see here an extraordinary effect in stabilizing, in green, the previously treated patients after enzyme therapy, and in naive patients achieving an improvement in the involvement of the bone marrow from diseased to mild or very mild. And bone crises, which were rare, nearly 400 patients, the bone crises also decreased on this therapy. So all the treatment looked successful orally, and we really wanted to make sure that patients would benefit in the long term and in the real world. And of course, these effects are still seen in patients who start the treatments in the real world, as written up here by Pram Mistry and colleagues with the company Sanofi by then, that has taken over the Genzyme legacy of eliglustat, called Sodelgra as the approved drug name. So it maintains the hemoglobin, and it has all those effects that you've seen here on the blood and the viscera, as you might expect. And also, it seems to have a good effect long-term on symptoms in the bone, in the skeleton. So, quality of life improves naturally, and patients remain well if they've already been treated with therapy. So this, I'm not here to advertise the drug. I'm saying that we have something and is effective. But the skeletal complications are there. And neurological disease varies across the world. In Europe and certain parts of North America and so on, where the literature is dominant, most of the disease is not neurological. It affects the skeleton if neglected, comes to light later, and in the viscera and of course the blood system. But neurological disease in other parts of the world is far more common than realized. And Gaucher himself never knew this. In fact, nobody really realized this in the West. So all these other features we had begun to understand, but we didn't understand the manifestations of the condition affecting the neurological system, as was understood from those early studies by Nilsson and others in northern Sweden. And the neuronopathic Gaucher disease, as it's known, is classified. There's the acute form, and then of the chronic form that was first described. There's aggressive systemic form with massive enlargement. There's a very rare form associated with cardiovascular calcification, as yet not understood. And then between the two of A and B is the Norbottnian type. When you look at genotypes for Gaucher disease, it was always dominated with the milder genotypes, which were recognized in southern and northern Europe. But if you go to the east, you go to parts of eastern, southeastern Europe, to China, southeast, and Southeast Asia, northern Sweden, you will come across patients who have a genotype that favors neurological manifestations, L44P homozygote, the one that is found in northern Sweden in particular. So here's the distribution. The non-neuronopathic Gaucher disease is found in the Middle East, Oceania, mostly in Europe and the USA, but in Africa, other parts of Asia, Indonesia, India, People's Republic of China, Korea, Japan, then we see something very different. And where the patients come from countries where there is nothing, like Pakistan, these are images given lockdown in 2020. Jim Shaman, we went there for a week. And these children have no chance of getting therapy, either enzyme which would help, or indeed a therapy for the brain. No access at all. And that is the responsibility for us all. I'll come now to something, a substrate reducing inhibitor, venglustat, which you've heard about, is not approved in any countries, and it enters the brain. And it is under as a drug once daily in very small quantities. And there are trials at the moment in Fabry disease, in late onset forms. But of importance, a phase 2 LEAP study in type 3 Gaucher disease, as you will all know and want to follow. And I can just share some of the 1-year data of this. There are 2-year data coming through. The trial included just a small number of patients with type 3 Gaucher disease that you know. They had to be stabilized, as it were, from the systemic viewpoint for at least 3 years with an enzyme therapy at a stable dose and be constant from the spleen point of view. They had to at least have a gaze palsy to indicate the neurological manifestations. And these are the criteria in their peripherals to see if in humans, if you gave the drug by mouth at the dose given, 15 milligrams per day, once a day, would the drug get to the appropriate concentrations in the plasma? And the answer is, after a few days, yes. And here are the range of activities. These are the sort of concentrations you're looking for to reduce the amount in the CSF and also in the peripheral blood of the activity of the enzyme that makes the glucosylceramide and, as it were, have the effect in the brain that you could in the plasma. And when you measure in the plasma and the cerebrospinal fluid the concentration of what's stupidly called GL1, but is glucosylceramide, and what's, if I may say so stupidly, called lyso-GL1, which is psychosine or glucosylsphingosine, which is the indicator of probably one of the active compounds related to this condition in the brain, you can see that over a year of exposure, in all but one patient, these values came down markedly, as it hoped for. And this is as expected. Well, it was well tolerated. There were no abnormal effects really to worry about, frankly, in the primary endpoint. There was a significant slight improvement in a score that's given to patients with ataxia, a little bit unsteady on their feet in gait, speech, and movement. And these tests showed on the whole either stable or a slight decrease in the patients who were exposed to the drug in the medium to long term of the 1-year therapy. This is very encouraging. But what was perhaps most encouraging were some refined measurements done by what we call brain, looking at connectivity, what's referred to as resting state networks, which identifies areas of activity, metabolic activity in the brain that are connected, anatomically connected. They're distinct, but connected in function. And we compared that also with very careful volumetric measurements to a very fine level in the brains of those patients. And here we show the values, including the patients that received the drug. One patient had some metal work and could not go into the missions here. You can see that there was these colors show a connectivity. The yellow colors show a connectivity between the regions of the brain that were studied. So at week 26 to baseline, and then 52 compared with baseline in the bottom panels, you can see that the resting state networks increased in nearly all the patients. Blue is less connectivity, green is no change, yellow is the enhanced value, which was in the majority. And then you can see this here, when we measured the brain volume, all but one patient increased. But in one patient who was not getting the drug, for one reason or another, never actually established, the whole brain volume decreased. So these values indicate greater coherence of the brain between the posterior and anterior aspects, the regions of the brain that enable transfer of information to be achieved as part of the way the brain functions as a supercomputer. And these are encouraging. They're not an endpoint for a clinical trial to get approval, but they are encouraging that there are possible recovery effects as well as stabilizing effects of this therapy. That's something I'm saying, not something that the company would say. I mean, I'm not trying to sell this drug to you or anyone else. I wanted now just to say very quickly what's going on before Professor Carlson later talks about this. Just to summarize, there are two forms of gene therapy, essentially, in this disease. And one of them, pioneered right from the beginning in the 1990s by Professor Carlson, was stem cell gene therapy. Here, retrieving bone marrow stem cells, correcting them with a normal activity of the enzyme, rather than using cells from a donor, which is very difficult, because there aren't many donors available for patients with this condition, and reinfusing the cells of the own person that should be acceptable for transplantation after conditioning into the patient, and seeing the effects that we would hope very much would provide additional enzyme that eventually can be transferred, as you will hear, by various means into the brain tissue. And this has been driven by the company AvroBio that bases its work on Professor Carlson's discoveries and recent refinement of the gene therapy mechanisms with a huge amount of sun. The other initiatives are considered, and these are some studies from another company not dealing with Gaucher disease, but just involves adeno-associated viral mediated gene therapy, which I'm not myself involved with in Gaucher disease, but in another condition. And this is what I mean, where you, as it were, inject a fairly harmless vector, which either finds its way to the sites, transfuses without going, we hope, to integrate into the nuclei of cells, and will get secretion of an enzyme, one hopes, in the appropriate place. Now, if you're going to deal with the brain, you would need to find a delivery that was effective for brain delivery. And this is a contentious area, either directly into the brain or into the cerebrospinal, a gene therapy. Adeno-associated viral mediated gene therapy is being explored by companies at various levels of expertise, and I'll come to that in a moment. Perhaps the most advanced gene therapy at the moment is that of AvroBio, which you'll be hearing about. And there's studies in patients with type 3 disease ongoing at the moment, and we'll be hearing about those at some point soon from that company, and no doubt from Professor Carlson. Freeline is a UK company which has a Gaucher type 1 clinical stage biotech program. And that involves, that program from Freeline involves, because of the work that's been done by that company and the colleagues involved in it, in hemophilia, which has been quite successful. One of the hemophilias, particularly hemophilia B. And so they've modified the enzyme here to be very stable, and hoping to get large excess of protein in the plasma for uptake into the peripheral tissues to secure a once-only therapy, liver-directed, for type 1 Gaucher disease. And a company in the United States also using AAV therapy, this is Prevail Therapeutics, was bought over by Eli Lilly 2 years ago, approximately, and uses a particular well-known gene therapy with intravenous injections for either Parkinson's disease program, challenging one, and also the very challenging acute neuronopathic disease in very young infants, an incident disease, really, a very great rarity, but does enable them to do those studies. We have no printouts, no data on these, but just to say, here at least there are more, 3 at least gene therapy programs in the clinic for different types of Gaucher disease, 1, 2, and 3. This is really remarkable. There are also additional substrate biosynthesis inhibitors. Takeda has published, Hansarts in the Netherlands, as a Pharos, has got a novel amino sugar, which is brain penetrant. So much going on. I just wanted to say these are encouraging times. I don't have enough time to talk about my own work. We do have 3 programs of work, 2, one in the lab, 1 genetics of Gaucher disease internationally, and 1 involved in clinical bone disease. And of course, work that Amy Donald is going to be doing and is doing from the Gaucherite program, which you'll be able to talk about and update, but which is now being. But I never think of the future. It comes far too soon, and we should be optimistic and very much hope that one or more of these programs will take us to yet another step in this journey of such a long time, and yet, on the whole, step by step, extraordinary progress. So thank you very much for inviting me again.